This is the story of how a scientist named Dennis Gonsalves saved the papaya production on the island of Hawaii with a crazy idea. In the 1980s, the papaya ring spot virus was slowly working its way through the Hawaiian Islands. And traditional plant breeding, traditional agriculture had no solutions. There were no known sources of resistance to this virus. Cutting out the diseased plants, traditional agronomic solutions didn't work either. So scientists tried a radical, long shot, literally, approach. They turned to a brand new technique that allowed them to move genes from one species to another, creating new sources of genetic variation. Inspired by work in tomato and tobacco, the researchers hypothesized that they could introduce DNA from the virus into the papaya and that it might work something like a vaccine, somehow protecting it from the virus. This is really crazy because papayas don't have immune systems like we do. But it had worked in tomato and tobacco, and it might work for papaya. They turned to genetic engineering, right? And a transgenic organism, a genetically engineered organism, is one that has genetic materials from a different species. How do they do that? Well, remember that a genome, right, an individual's genome, all of its genes, are made up of chromosomes. And the chromosomes, in turn, are made up of genes. The genes are composed of DNA. And DNA is DNA is DNA. If you take a gene from a fish and you put it into a banana, the cellular machinery of the banana treats it just like the other DNA in the banana and makes the same protein. It doesn't matter what organism you're in, it makes the same protein. And that's the key to why this works. So in the case of papaya, they wanted to move a gene that encodes a protein on the virus's outer coat. Papaya ring spot virus is a long, thin virus like this, studded with proteins, in the same way that our red blood cells are studded with proteins. That's what makes someone have an AB blood type. So the first thing they needed to do was find the gene that makes the protein on the virus's coat. Once they found it, they needed to package it up in a way that they could move it into the papaya. So here is the vector that they created, a ring of DNA that contained the virus's coat protein. But that's not enough. You have to know whether or not the protein arrived where you wanted it to, and for that you need a reporter gene. And in order to make sure it works once it's there, you need a switch gene to turn it on. So all of this is packaged together into a vector, which then has to be moved into the plant cell. Now that's a non-trivial problem, because plants have cell walls, and they're hard to get inside of. And once inside, you have to get all the way into the nucleus in order to reach where the DNA is. So how did they do it? The answer? They shot it in. So this is the modern version of a gene gun, and the older versions used a .22 caliber shell filled with vector-coated, right, DNA-coated tungsten beads that they shot into the papaya cells, some of them making it through into the nucleus and getting incorporated into the papaya's genome. It's a long shot approach, literally. Next, the researchers use a combination of plant hormones to turn this callus, to make it grow first roots, then shoots, and finally turn it into whole plants. Now, the researchers don't know, right? This is an odds game, right? The chances of getting the DNA into the nucleus are very small. And so they needed to screen many plants in order to know whether or not they had transgenic plants at all. Once they did that, they needed to figure out whether or not they were resistant. They didn't know whether or not this would work. So how did Dennis Gonsalves and his team figure out whether or not this experiment worked? Well, they ran an experiment. They put together control plants and transgenic plants, right? And what did they do to find out? Well, they infected them with papaya ring spot virus and watched to see which ones got sick. Now, what's your prediction? After 10 days, all of the control plants and most of the transgenic plants were sick. But there was one transgenic plant that was thriving, deep green, growing fast. It was not getting sick. And they watched and eventually began to trust that this plant was actually resistant. But what works in the greenhouse is not always the same as what works in the field, and the next step was to move out to the farmers' fields. But before they could do that, they needed the transgene in a commercially appealing variety, right? Resistance is only one quality that farmers and consumers care about. So plant breeders worked to put the transgene into desirable varieties, and the result was 2 different cultivars. Sun Up has pink flesh and 2 copies of the gene. Rainbow has yellow flesh and only 1. It was made by crossing Sun Up with a popular variety in order to produce the different variety. So, does 1 gene versus 2 make a difference? Well, here's how they found out. They went out to the field and planted several different things in the field. Here are the different varieties that they planted. 2 different Sun-Ups, right? Rainbow, like we just mentioned. The transgenic line itself, not good production qualities, but at least we knew it had the transgene. And then 2 traditional varieties. And pay attention to what's over here in parentheses. That tells you how many copies of the gene it has. So the traditional ones have none, right? And the transformed ones have 1 or 2 copies of the gene. So what happened? In January 92, they planted these different varieties in one of the most infected areas in Hawaii and watched to see what would happen. After just 6 months, almost all of the traditional varieties were infected, right? Nearly 100% infection rate. But down here, stacked on top of each other, are all of the transgenic plants. The Sun-Ups, the Rainbows, the transgenic line itself, none of them infected. And so what's the conclusion? This wild idea of Denis Gonsalves's had worked. The crazy thing is that it was decades before scientists understood why.